Hello everyone, happy February. Today we are going to talk specifically about the idea of genetic variety and how do we actually get that? Trying to address, a simple question as, why do siblings not look exactly the same? So we're going to get started and kind of learn a little bit about that. We're going to do kind of a fun fruit fly activity. And as you can see on the screen, just making sure that you get down the initial notes and then putting your info on the class data slide so that we can analyze it next class. So I'm going to go ahead and click into entry number 16. And so the first thing that I'm going to do for those who are in class is just kind of recapping, right? We left off last class learning about how our sexual life cycle is a process of kind of going back and forth between meiosis and fertilization. Meiosis is the process you see in the picture here on the right, but that is a special process by which cells, our gametes, our sex cells, are created to contain half of the normal genetic material that make up all of our other cells, with the idea that they need to contain half of the genetic material because they're going to come together with another gamete and through the process of fertilization create a zygote. So these special sex cells need to have half the genetic material so that when they come together, we get back up to that full set. So we learned a couple vocab terms last time. Those gametes or sex cells are haploid. They have half of the total genetic material, or kind of one set of 23 chromosomes. And then they will come together and form a zygote, which is diploid, or having two complete sets, that total 46 number that we've learned about. So I'm just going to open it up to the class to kind of review that, and then if there were any specific takeaways from the video that they chose to watch and learn a little bit more, just kind of picking out something they put on their B page to share in the chat. So that's what we're going to start with. And then we're going to go into the idea of genetic variety and how do we actually get that. So I'm going to pose the question to the class, right? If everyone gets half of their DNA from their mom and half of the DNA from their dad, why don't all siblings just look the same? And I'll pose that. You can kind of see, hopefully you know who these sets of siblings are, otherwise I'm very old. But you have the Hemsworth brothers on the left, you've got the Wilson brothers on the right, and you've got the Olsen twins in the middle. Or I guess siblings, right? There's the pair of twins and then their sister in between. So why don't all siblings look the same? If they're getting the, half of their genetic material from the same sets of people, why is it that they don't look exactly the same? That's kind of the question that we're going to pose today and learn a little bit more about. So in your notebook, I want you to go ahead and open up to a very fresh page. We're going to start entry 16 today. And this is something that we're going to continue on into next class as well. But our essential question is, how do meiosis and fertilization lead to genetic variety? So we're going to learn about how we get that difference, that variety, so that everybody isn't exactly the same. Okay. So if you haven't gotten that down, pause, and then once you do, go ahead and push play again. Okay, so in order to address this particular question, we need to learn a few more vocabulary words. And I'm sure that you're thinking, I've already learned a whole bunch, but now we're going to learn a couple more because they're necessary for us to understand and answer the essential question as well as do our activity in a little bit. So one of the words that I would like you to write down is kind of the info on the left. So pause the video now and get down the information right underneath your essential question. Go ahead and write the definition for an allele, and then even this bullet point underneath is kind of worth writing down. So pause the video, get that down, and then when you have it, go ahead and resume and I'll talk about it. All right, so an allele is a version of a gene. We learned previously that a gene is a chunk of DNA that codes for, a specific protein. So in my little picture example here, I have, this location on the chromosome, and at that spot is where this particular gene is located. An allele is a version that you happen to have there. So alleles can have 2 or more options. So it's kind of like the variety that exists in alleles. Some people have one version, some people have another version. And if there's more than 2 options, you can have any one of them. So for example here, the chromosome on the left has the allele for pink flowers, and the chromosome on the right has the allele for blue flowers. Just like if, this was the gene that controlled our eye color, right? There would be a certain code of DNA that would make our eyes blue or green or brown or any of the many, many shades in between. And as you can kind of see, I tried to highlight the fact that you get one allele from each parent, right? This is a pair of homologous chromosomes. So these could represent 2 chromosome 1s. And so you as the child have kind of one pink flower allele in this situation and one blue flower allele. You got the pink one, let's say from dad, and you got the blue one, let's say from mom, right? And so that's your particular combination, knowing that you got one from each. And in some instances, they might be exactly the same. If this particular plant's parents, so to speak, both had only pink flower alleles, then that's possible. Or only blue flower alleles, that's possible. And so we're going to talk a little bit about that too. All right. So the other thing to note is that there are different alleles or versions of a gene that you can have. And they don't always play equally nice. It brings about the terms dominant and recessive. So go ahead and pause the video right now and see if you can write down what I have for definitions there. Alleles are usually either dominant or recessive. So write them down, and then when you're ready, hit play and I'll talk about it. Okay. So alleles being dominant, if the allele is known to be dominant, then it is the one that we see in terms of a phenotype. It's the thing that we can see whenever it's present. And we usually show it in the form of a capital letter to show that it is the dominant letter of the 2. And so that's why this picture kind of has the capital B bullying the little b, and why in this particular case we're showing that the pink version is dominant. So if you get that big B or the dominant allele, no matter what, that is the trait that you're going to see. Recessive is an allele that will only ever show itself if there isn't a dominant one around, right? So we usually denote these with a lowercase. So in our example, blue flowers are getting that lowercase letter. So even if this person has one little b or blue flower allele, it's masked by the dominant one. The only time we would ever get blue to show itself is if there was not a dominant one around. So here's kind of what that looks like in practice. So I'm going to describe it, and then I'm going to have you guys pause and draw it into your notebooks. And I did include a picture of my own notes if that's helpful for you. You can kind of see that. But I've laid out 3 possible situations. So one situation that is possible is that you get one big B from mom and one big B from dad. Both of the parents happen to have pink flowering genes. So your genotype, if we remember, genotype is the combination of alleles. It would be big B, big B. And since you get one from mom and one from dad, you're always going to kind of have this 2-letter combo. And then we go ahead and get to a second situation where you might inherit one pink flower allele from one parent and one blue flower allele from the other parent. So one of them is dominant and one of them is recessive. So you end up with this big B, little b situation. And then finally, there's the chance that you might have, sorry about that, the final situation is if a person has inherited 2 little b's or both parents donate a blue flowering allele to the offspring. So their genotype would be little b, little b. So you can see how for any given gene that has, 2 options, there's kind of 3 choices they can contribute to the offspring. And so those are what we call genotypes. That's the genetic code that a person has inherited for that particular gene. And if we remember the phenotype, pheno means physical. It's what that looks like. So you can see if it has a capital B, that means that that trait is, or that allele is dominant. So that's what we would see. It doesn't matter if there's 1 or 2. Either way, if it's present, then you're going to see that particular trait. And the only way you will ever see the recessive trait is if the dominant allele is not there to mask and overpower it. So that's why we end up with blue flowers only in the little b, or double lowercase situation. Okay. So now that I've talked about it a little bit, I want you to pause and see if you can reconstruct this in your notes page. Just kind of highlighting what you see here, as well as those vocab terms genotype and phenotype as well. If it's helpful to check the PowerPoint, you can check my notes to see what I did, or construct it yourself. All right, so hopefully you got a chance to record that info, and if you haven't, please make sure you pause and do. But now we're going to come back to that question. So what does that vocabulary that I just talked about have to do with variety and why you don't look like your siblings? Well, I have the answer for you. And so alleles, those different versions that come from mom and dad, are packaged randomly into gametes. So in a mom, the mom's alleles are packaged, they're lined up and separated and individually packaged into egg cells. And for a man, that same process of meiosis, right, occurs to create sperm cells. And so kind of the random lining up of all the chromosomes and then splitting them up and separating them sorts them into random combos of eggs and sperm, and that creates variety. So think about it, I don't know, like a random, bag of Skittles, and you have one full bag, you kind of dump it out on the table, and your job is to just kind of line them up in pairs and then split them into 2 equal groups and then split those into 2 equal groups. So you end up kind of with 4 equal piles of Skittles. And depending on how you randomly line them up and separated, right, each one of those little piles is going to look a little bit differently. And if you, pushed them all into the center and did the process again, you would probably end up with different situations than you did the first time. So all of that kind of mixing around leads to increased variety. And so you really never know, any given sperm or any given egg kind of ends up with a different combination of traits or, chromosomes that will lead to traits. They're not all identical. So please pause the video for a moment and kind of underneath where you drew the pictures of the genotype and phenotype chromosomes, I want you to get down the text here on the left. I know it seems like a lot, but they're kind of important notes to have. So just pause, write that down, and when you're ready to continue, go ahead. Okay. So now that you have this down, kind of understanding that all of the eggs and all of the sperm are going to look a little bit differently, like that is one reason why you don't look like your siblings, because the sperm and egg that made you are different than the sperm and egg that made a sibling. Even though they're coming from the same people, they have a little bit different chromosome combinations. So when you put those different chromosome combinations together, then that leads to variety. And so kind of to highlight that point, like I said, two gametes with different genetic info coming together, right? The one magical sperm that makes it to the egg at any given moment, that random chance of those two sets of chromosomes mixing to make you, it really creates a super unique combination of genetic material that no other individual has. Even close siblings wouldn't have that same combination. And so just understanding that that's another place where variety comes in. So kind of as your last note on your A page, go ahead and pause and kind of jot down this fact here, that during the process of fertilization, two gametes that we've already learned are unique from each other are now coming together in a unique combo from both parents. Okay. So the last thing that I have for the A page is just kind of review. So I did put a quick little video recap that I'm going to show in class. So if you go to the PowerPoint, you can watch this nice little tiny review that kind of goes back over the vocabulary so that it kind of sticks a little better. And on the following slide, I have a picture of my own notes page. So if you wanted to pause and zoom in or look back at this as a guide, you could. You don't have to by any means. Yours might look totally different, but that's just kind of what mine ended up looking like. Okay. So with the second chunk of class, we're going to end up putting all of this into practice. We're going to do a little fruit fly activity to highlight the process of that genetic variety and how that happens. So we're going to use Drosophila melanogaster, aka the fruit fly. Super common model organism in genetics experiments. In graduate school, I actually worked in a fruit fly lab for a while because they are, scientifically awesome for a lot of reasons. And if you're wondering why scientists look at fruit flies to try to learn anything about humans, I put this little video here. But some of the key pieces include the fact that it has only 8 chromosomes. So instead of having 23 pairs totaling to 46 like we do as humans, they have 4 pairs totaling up to 8. Which is a lot less genetic material to mess around with, so it's easier to do things with. They have a similar XX for female, XY for males, like we do as humans, so we can kind of take information from there. And we actually have a lot of similar genes, or genes that do similar things between humans and fruit flies. So if you're curious, there's this cute little short video that kind of talks about the significance of fruit flies. And they're small. You don't feel bad killing them. They don't live very long, so it's easy to get lots and lots of generations. All good things. You can see on the right here a karyotype, or that picture of chromosomes for the fruit fly. You can see the 4 different pairs. So there's one that kind of swings to the left. There's one that kind of swings to the right. The 2 the pair that basically looks like a dot, and then XX on the left, and XY, the Y kind of has a hook on it on the right. So that's what a fly karyotype ends up looking like. And if you remember, we drew our own in the last entry. So that's kind of what that looks like. Fruit flies are awesome because there's a ton of different traits that you can actually look at and assess. So whether it's like their antennas, we can look at their wings and what they look like. We can look at their legs. We can look at their body colors. They have fairly complex eyes, so we can look at different color eyes. We can look at different sized eyes. You can look at a lot of different traits with fruit flies. And so I want to highlight a couple that we are going to highlight specifically today. We are going to look at eye color, wing size, and eye size. So in our case, we have eye color, and we have two options that we're working with today. Red eyes, which is the dominant, right? That's the normal, so to speak, color. We also have brown eyes, which is recessive. So if we think back to our pink and blue flower example, we would only get brown eyes if the dominant allele, the red one, wasn't there. Next, we're going to look at wing size. So they either have long wings, which is dominant, or they have really short wings, which are recessive. And then eye size, they can either have big eyes, which is dominant, or they can have small eyes, which is recessive. And just kind of highlighting the vocabulary, again, genotype are the lettering, phenotype is what we see in terms of the different versions. Okay, so you don't have to write this down. I just wanted to go over it so that you would be familiar with some of the things that we're using in the lab. Okay, so here is the actual activity, and I should also highlight the fact that you can tell the sex of the fruit fly by looking at its abdomen. So females have kind of bigger, longer, striped abdomen, and black tips are usually on the male, so they have kind of a darker black abdomen. And if I hop back to the previous slide, you can see it in actual flies here, the female on the left and the male on the right. But our goal today is to kind of do the process of meiosis and fertilization in order to see what kind of offspring two flies would make. And our parent flies, so to speak, are both going to be big R, little r for eyes, big V, little v for wings, and big E, little e for big eyes. So both parents that we are crossing today have red eyes, long wings, and big eyes. But they also have a recessive allele that we can't see right now because there's a dominant allele present. But we're going to see what happens if we cross two parents that have that particular genotype. So I have the activity linked on the left, and then a coin flipper, and I'll explain where that comes in a moment. Okay. So here's the activity that I'm going to roll out in class, and as we do it in class, I'm going to actually put people in breakout rooms in partners to work together, and each partnership would have their own slide. So if you are watching it later, you can kind of observe and or pick up a slide that no one has picked up. So here are the instructions, and I'm going to have to walk the class through this as we do it, but we are going to begin with two parent flies that have the exact same genotype that I mentioned on the other slide. So they are big R, big V, big E, little e. So that part we already knew. And then I have one boy, so XY as the dad, and one that has XX, the mom. So I have two parents. Those are my parents and those are their genotypes. On the actual slides of the activity, they look a little bit like what you see on the right here. You can see how there's the chromosome that contains the R gene, there's the chromosome that contains the F gene, the chromosome that contains the E, and then the sex chromosomes. So you can see how both mom and dad have those 4 pairs of chromosomes, and we can see the different versions that they have. So that's where the big R little r is coming from. The big F little f is coming from. Big E little e, and then for dad, it's XY, and for mom, it's XX. Just kind of highlighting where those came from. So what you're going to end up doing is using a coin, or if you want, you can use this electronic coin flipper. You're basically going to flip a coin to determine which of the 2 versions of a gene make it into the gamete. So if dad here has big R, little r, I'm going to flip a coin to say, okay, this one goes into the sperm. And that's where the flipper comes in. If it lands heads, then the dominant allele makes it into the gamete or sperm. If it lands tails, then the recessive allele is going to make it into the gamete or sperm. And that's going to be true for each flip. You're going to flip 1 coin for the R gene, 1 coin for the F gene, 1 coin for the E, and then the last one is just going to be for the sex determination. And I just said heads is X, tails is Y. So if I click into the coin flipper just to kind of show you what it looks like. Takes a second. I just used a penny, but you are more than able to use whatever you want. I'm going to flip 1 single coin because I want to kind of walk through each gene separately. If you click here, I could say I would like to do a Swiss franc or something else. So many choices. I'll do a Swedish krona because I'm Swedish, so why don't we use that? And then I hit flip coin. Voila, it's heads. And then I could do it again. Flip. I think it was heads again. Oh, now it's, I think that's tails. The key part of this is that you need to know which is heads and which is tails. So you're going to walk through and you're going to flip the coin until you have done it a total of 4 times. And when you do that, then you end up with your dad's gamete or the dad's sperm. Like this would be the sperm that contains this genetic material. And each group might have a difference in what they end up with, right? Because it's totally random. You're then going to repeat the process for the mom's egg or the mom's gamete. So when you end up doing this process, you'll have 2 sets. You will have dad's gamete that you've determined from kind of coin flipping for each gene, and then you'll have mom's gamete that you've determined from coin flipping. And remember, moms can only give X's, so a mom cannot give a Y. You cannot have a child that is YY. But you're going to then fertilize. So we're going to put those together and make a fly baby, so to speak. So if I put these 2 together, I end up with big R, little r, because I had a big one, then a little one. Big F, little f. Big E, little e, X, Y. Congratulations, it's a boy. So that's my kid right there. That's our fly baby. And so we're going to record this genotype and we're going to actually create a fly based on what it looks like. We're going to do this for a total of 2 babies. So we're going to repeat the process 1 more time and make another baby, see what we get, and record our data. So that's the instructions piece. I'm going to walk you through the slide that you will be assigned because it looks a little intense. All right, so what you're going to do is each group, Again, let me just kind of walk you through it. Each breakout room is going to have a slide like this. You're going to end up putting your names right here. So on the top row here, like first person, second person, you're going to be working in partners. Along the side are the same chromosomes that I showed you before. So that's just showing you mom and dad's karyotype that were, big R, little r, big F, little f, big E, little e, and then mom versus dad. That is the exact same for everyone. So once you go through the flipping process and you get your genotype, you're going to put that here. So this is the combination that I ended up with after I did all of my random flipping. You might do the same thing and get another. Let's say I get big R this time, and then little f, and then big E, little e, and it's a girl. So that might be an example of a different combo that I end up with when I do this the second time around. So once you have the genotype, you are going to actually make your fly. So I know big R, little r. Big R is dominant, and that is red eyes. You can use the chart at the bottom to help you if you don't remember which trait is which trait. So if it has a capital R, then it's going to have red eyes. Okay, so I know it's got red eyes. And then I also kind of need to look at the size. Are they big red or little red? Well, my fly has a capital E, so it's going to have large red eyes. So I kind of come over. Oops. I'm going to click on the head with big red eyes, and I'm going to drag it on over to my fly. Check. Other things, I know it's a boy, so I need to pick the male abdomen. Kind of bring that over. And then in terms of wings, I have a big F and a little f. So it's going to have long wings. Voila! So that's what I end up with. If I wanted to say that my other one was big R, little f, big E, little e. Let's say that this is my other one. It's going to have red eyes again and large. So this one would have large red eyes too, except the difference is it's going to be a girl. So it has a female abdomen and 2 little f's. So this one actually has short wings. Voila, I've made 2 little flies. So this is what my 2 potential offspring would be. You can name them. So where it says offspring name, you can name them if you'd like. Please make sure that that is school appropriate. So once you get that piece done, you're going to scroll all the way to the bottom of the slideshow after all of the groups, and we have our class data. So you are going to add your information to this chart. For example, I had one that was a boy and one that was a girl. So girl, this is the symbol for girl on top, symbol for boy on the bottom, and you're going to add to the list. So if it already said 5, you're going to make it say 6 or whatever you need. And then I need to figure out where my flies were. Which ones did I have? And in this case on the right, the sex of the fly doesn't matter. It's just looking at eye, color, size, and wings. I had one of these, big red eyes with large wings, and I had one of these, big red eyes with small wings. And so the goal is to have the whole class add in their data to this final slide, and then we will analyze it next class. Okay. And I know that this is going to take a chunk of time, so we'll see how it goes in class as well. And so kind of wrapping it up for the day, we've kind of learned a little bit about what alleles are, the terms genotype and phenotype and how they play out, and then started to work a little bit with fruit flies. And so the homework piece is to just make sure that you get your team's slide done and data added to that class data set. And we'll pick up again on this entry next class. All right, thank you.